Now the guidance documents on genotoxicity are listed here. You go through these documents so that you get more information on the control of impurities, what are the levels, what is meant by threshold of toxicological concern, and so on and so forth, and several other terms. We will be now focusing on the operative part of M7 guideline. Operative part meaning, if you see here on this slide, the points 1 to 4 are related to introduction, general scope, principle, and the administration about products and marketed products and new applications and all that. Whereas point 5, 6, 7, 8 are related to operative part, and that is what we will discuss in detail. This is about impurity assessment. Then the classification of these impurities based on the hazard they are likely to have, and then characterization of those in terms of what is the allowable daily intake or acceptable daily intake in case of that impurity. So that means kind of setting a limit for daily exposure, and then about controlling that impurity in drug substance primarily, and also in drug product. But in drug product, the impurities to be considered are mainly degradation products, and if there are any drug substance and excipient interactions. And then documentation. We'll briefly talk about documentation at the end. So the assessment impurity of impurity starts with the question, what impurities need to be assessed? Then hazard, how do we assess these impurities? How do we know, or how do we find out whether impurity is likely to be or potentially mutagenic? Then in risk characterization, we'll see what are the acceptable intakes. And in control, as I talked earlier, we'll talk about the controls, that various types of controls that we can have. So impurity, can be classified. We have seen it in other discussions as organic, inorganic, or residual. We have seen it in discussion of Q3A, Q3B, and Q3C is on residual solvents. So we are familiar with this class of these three broad classes of impurities, and from where what is their likely source in the drug substance. This is based on the drug API process, drug substance process. Now, when we know about an impurity, if we write the balanced equations for the API synthesis right from the regulatory starting material, and we also write the possible impurities that may be generated stage wise, considering the carryover from previous step. If we do this detailed exercise, the impurities meaning we will consider overreaction, underreaction, side reaction, and also, as I said, reaction of the carried over impurity in that reaction step, in those reaction conditions. Now, when we detail out all these impurities, if we look at the structure, there are certain alerts, which we'll see it on next slide, which are those structural alerts. Those alerts indicate, or that substructure indicates, that it is likely to be, or it is a potential mutagenic impurity. This concept was introduced by John Ashby in 1985. So here are some examples of structural alert. N-hydroxyls, hydroxylamines, then N-acylated aminoaryls, then alkyl esters, haloalkenes, and alkyl allyl groups with nitroso, nitro, the epoxides or aziridines, means three-membered rings. These are some of the examples of structural alerts. What we notice is these are highly reactive uh, functional groups which can react with the base pairs, adenine, guanine, thymine, uracil, nitrogen containing base pairs. They can transfer like um, alkyl esters. They can transfer the alkyl group on the to the nitrogen. So the structure of the base and changes. Now this change is called mutation, because the basic structure, the inherent structure of the DNA molecule is then undergoes change due to this type of reaction at the base pair. So all these are reactive molecules, and they'll be potential. If any of this functional group is present, then it is that impurity is treated as potential genotoxic impurity. Now this is a hypothetical structure, just collected or connected, written so as to include all possible alerts in one structure or in one graphic form. Now this is not a real chemical. Remember, it's just a hypothetical structure to explain all possible structural alerts in one go. The point to remember is all of these are highly reactive functional groups which are likely to react with DNA base pairs. Now, apart from these structural alerts and compounds containing structural alerts, there are certain cohorts of concern. Cohort is a class of compounds, a group of compounds. A group is called cohort. So aflatoxins, there are various derivatives of aflatoxins, A, B, C, and so on. And in B, there are various, again, B1, B2, up to B12. So there are class of compound, but the base structure is shown here. And if your impurity has structure similar to this base structure, then it is a cause of concern. Same way, azoxy. We carry out diazotization, so azo compound N double bond N is formed. It is likely that it may undergo oxidation in during the reaction, air oxidation or some such impurity, some such thing, or azoxic. So any azoxy compound formed of this nature is again cohort of concern. Same way, nitroso compound, N-nitroso compounds, either N-nitroso methylurea, methylamines. You know, at present, N-nitroso compounds, nitrosamines are causing wide